Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we have to wrap up this idea of phosphorylation, and we haven't talked about one, and that is photophosphorylation, photosynthesis. We're going to be talking about it today. Thank you so much for being here. I'm going to give you a quick overview here, a quick picture of what we're going to be talking about. Here you go. Now, what we have here are the basics of photosynthesis. On the left-hand side, you can see the sun, i.e. visible light. These are called the light-dependent reactions. And for the light-dependent reactions, we're going to need some light, we're going to need some water. And the whole purpose of the light-dependent reactions is to make ATP and reducing power in the form of NADPH. And that ATP and that reducing power is going to go to the Kelvin cycle on the right-hand side. And the Kelvin cycle is an example of light-independent reactions. And so in the light-independent reactions in the Kelvin cycle, we're going to, the plant or algae or cyanobacteria, the photosynthetic organism, is going to utilize CO2 in an anabolic process along with ATP and the reducing power, and make organic compounds like sugars. And so what we're going to do today is we're going to kind of get into the meat of what that means for both the light-dependent and the light-independent reactions. But for the light-independent reactions like the Kelvin cycle, we're going to keep it very brief. But before we get into the details, just a quick reminder, when it comes to visible light, we have the energy spectrum here all the way from the very low energy radio waves, so very, very large wavelength, long wavelength. And as we go from left to right, the wavelength gets shorter. The energy picks up a bit. And so you can see radio waves like your FM, AM radio, and you've got microwaves, the microwave that you cook your food with, the IR infrared rays, and then we get into the visible light, and that's the usable light spectrum for our photosynthesizers. And then once we get beyond that, still usable stuff. We've talked about UV light in the past for controlling microbial growth. You've got X-rays, very high energy, very important in the medical field. And you've got the gamma rays, and those gamma rays, extremely short wavelength, very, very high energy. And we're thinking nuclear reactions there. But our focus here really is on the visible light. And you'll notice for the visible light, on the left-hand side, we go from the reds into the oranges, eventually into the yellows and greens, and then we get into the blue and finally the purple or violet. And so when it comes to visible light, most photosynthetic organisms are going to be able to absorb energy at the ends of the visible light spectrum. So in the reds and the oranges and the blues and the purples, that's where they absorb most of the light, and of course they reflect the green light back. That's why if you have a photosynthetic organism that appears green to us, the color that we're seeing is based on the light that is being reflected off of that structure. And so when it comes to photosynthetic organisms that appear green, they're not really using a whole lot of light in the green part of the spectrum, with the exception of some accessory pigments that might be there, like the xanthophylls and the carotenoids that can absorb a little bit of that green light, but the vast majority is on the ends. So to explain this a little bit easier, I think we need to go to the whiteboard, and we're going to explain the light-dependent reactions there. So let's go venture off to the whiteboard. All right, so here we are at the whiteboard. This is the photosystems. We got photosystem 1 over here, photosystem 2 over here. Don't worry, I will get out of the way. But I thought it would be a great idea to start with photosystem 1. That's the easier of the two to explain. And so in the light-dependent reactions, we have a couple of photosystems working together that allows the photosynthetic organism to produce ATP and to produce reducing power like we talked about before. And that ATP and reducing power is then going to be used at the Kelvin cycle to store that energy if the photosynthetic organism ever needs it in the future, because the sun isn't always shining, visible light isn't always present. And so here we are, let's start with photosystem 1. And so basically what we have here is a representation of chlorophyll down here. It says photosystem 1. That's what the PS1 stands for. And what's going to happen is we're going to have some visible light. Let me draw that in right here because I forgot to draw it in. So we've got some visible light that's going to hit that photosystem, and it's going to excite some electrons. That's what we've got here. We've got excited high energy electrons. Those electrons, if you follow the dotted pathway, they're going to get excited, then they're going to come over here to the proton pump, where just like, or sort of like what we see with the electron transport chain, the proton pump creates a gradient that allows for phosphorylation of ADP into ATP. And so we get some ATP production from photosystem 1, but then that electron comes right back to photosystem 1. And so there's a couple important things that we need to know about this photosystem. First thing is that photosystem 1 is cyclic. It's a cycle, meaning the electrons get excited, they make ATP, and then they come back. And that photosystem 1 is in fact responsible for making ATP. Now on the microbiology side, not all photosynthetic organisms have both photosystems. In fact, it's somewhat rare. Now, so that's photosystem 1. It's a cycle, it makes ATP. Now if we come over here, I'm going to have to come off screen here so you can see this a little bit easier. Photosystem 2 is a different situation. And so right here is photosystem 2. We still have visible light that's exciting electrons from photosystem 2, which is what we have right here. The electrons, now we're going to follow the solid line. The electrons get excited and they go and they make ATP at the proton pump. But now look what's happening. Those electrons are not coming back. They're not coming back. In fact, they're going to go from photosystem 2 straight to photosystem 1. And when they hit photosystem 1, over here, I can come back on screen now, they're going to get re-excited, and then those electrons are going to be used to convert NADP+, that electron carrier, to NADPH. That's the reduced form. And so now we have reducing power. So what can we say about photosystem 2? How is it different? Well, if we come over here, first thing is it's not a cycle. The electrons never came back like they did in photosystem 1. So the electrons got excited, they made ATP. They went over to photosystem 1, got re-excited, make NADPH. And so they're making ATP plus reducing power in the form of NADPH. But there's something else here to consider, and that's right here. If photosystem 2 continually loses electrons, you could imagine a situation where in fact there is no more electrons available for photosystem 2, and so electrons have to be replenished, and the organism needs an electron source. And so for a lot of our land plants, for our cyanobacteria, for our algae, that electron source is actually water. It is not the sun. It is not visible light. The electron source is water. Water provides the electrons along with some enzymes that allow for this process to happen. And when water is used as an electron source, the organism releases oxygen as a byproduct. It's a waste product. So these are the light-dependent reactions. And so we can add one more thing to photosystem 2, that it uses water as an electron source, and it is producing oxygen, unlike photosystem 1. In the next video, we're actually going to talk about organisms that are photosynthetic that don't use water as an electron source, and we'll start giving them some names as well. All right, so now that we know that, we know what's being produced in the light-dependent reactions. We have our ATP and reducing power. Those things are the usable things from the photosystems, okay? So when it comes to light-dependent reactions, the ATP and the reducing power are the usable things. The oxygen is usable to us, but not immediately useful to the photosynthetic organism. It's a waste product for them. Now that we have those things, if the photosynthetic organism wants to store this energy, well, the way to do that is in the Kelvin cycle. All right, so now that we have the light-dependent reactions and we understand that we're getting some ATP, we're getting some reducing power, but what if we want to store that? What if the photosynthetic organism wants to store that energy? Well, in the light-independent reactions, we've got the Kelvin cycle here, and we can see that it is all kinds of stuff going on. And again, just like we did for the Krebs cycle, we're not explaining every single one of these steps, but there are a few key points on the left-hand side that I want to make, and that is the Kelvin cycle here. Now this thing, what's happening is it's actually turning 2 times to make a single 6-carbon sugar. And so what's happening at the top, you see that there's 3 CO2. And so to make a 6-carbon sugar, you're actually going to need to have this thing turn twice, and you're going to need 6 CO2. And then you're going to see that there's 6 ATP being utilized on the right-hand side as well as 3 on the left-hand side. Again, it's going to turn twice, and so it's actually, we need 18 ATP per sugar molecule, and then the reducing power, you can see that we use 6 NADPH on the right-hand side there. Again, we double that because this thing has to turn twice. And then for a glucose, then what we really need is we need 6 CO2, which glucose is a 6-carbon molecule. That makes sense. 18 ATP and 12 NADPH. All right, everyone, hope you enjoyed that. That is photosynthesis, photophosphorylation, the light-dependent and the light-independent reactions. In the next video, we are going to be talking about a lot of different ways that organisms obtain energy and use terminal electron acceptors. We're going to talk about where we find them. And so we'll really be able to tie all this together. As always, thank you so much for being here. We'll see you next week.